parenchyma, sclerenchyma, meristematic, epithelial. Biology textbooks often hit you with a dense wall of terminology and complex cellular structures all at once. Before you can accurately look through a microscope and identify these structures in a lab, you need a mental framework of how they actually relate to one another. We are going to build that framework right now. Grab a large blank sheet of paper and 3 different colored pens. We will start by establishing a clear spatial rule. Dedicate the entire left half of your blank page strictly to plant tissues. Translating these abstract textbook paragraphs into a physical, color-coded spatial map helps you build the visual memory required for high-pressure exam environments. Plant architecture is defined by a single division. Is the tissue actively growing, or is its structure fixed? Draw a branch splitting plant tissues into 2 distinct categories. Write M for meristematic and P for permanent. Meristematic cells are the rapid dividers. To maintain that speed, they have thin cell walls and lack large internal storage structures called vacuoles. Permanent tissues have lost the ability to divide. Because their shape is fixed, they develop thick, rigid walls and large central vacuoles to store sap and support the plant's weight. Meristematic growth occurs in 3 specific locations on the plant. You can map them using the acronym ALI. Apical meristems sit at the root and shoot tips, driving vertical length. Lateral meristems push outward from the trunk, increasing the plant's horizontal girth. Intercalary meristems sit at the nodes, extending the stem between the leaves. By directly attaching these growth terms to specific directional arrows on your map, you clarify the distinction between primary vertical length and secondary horizontal girth. Transitioning to the permanent tissue branch, we organize the physical shapes into the PCS framework: parenchyma, collenchyma, and sclerenchyma. Parenchyma acts as the living storage unit. Sketch a thin-walled circular cell. In aquatic plants, like the lotus, these cells adapt into aerenchyma, specialized tissues that trap air to provide buoyancy. Collenchyma provides the flexible support system. Draw a cell and heavily shade the corners. This specific corner thickening allows stems and leaves to bend in heavy winds without snapping. Sclerenchyma forms the rigid barrier. Draw a thick, solid block here. These cells are dead at maturity and packed with lignin, a tough polymer that hardens structures like the outside of a coconut shell. Labeling these specific shapes as living or dead right on your map serves as a practical guide when you are analyzing real plant cross-sections. Now shift your attention to the right side of your paper. We build the animal tissue matrix using the structural acronym NCERT. E stands for epithelial tissue. This is the protective shield. It forms a continuous layer over your skin and lines your internal organs to block pathogens. M is for muscular tissue. This is the mechanical engine, responsible for contracting and relaxing to drive every physical movement your body makes. N is for nervous tissue. Concentrated in your brain and spinal cord, this is the signaling network that receives external input and fires electrical signals to trigger your reactions. C stands for connective tissue. This is the internal transport and binding system that physically links all of these separate organs and systems together. Visualizing these 4 separate tissues as a coordinated mechanical team transforms isolated vocabulary words into a logical, interconnected biological system. Let's create specific sub-branches for the muscular tissue to differentiate the 3 levels of physical control: striated, smooth, and cardiac. Striated skeletal muscles operate under your voluntary control. You decide when to reach out. Smooth muscles operate your internal organs through involuntary automation, pushing food through your digestive tract without your conscious input. Cardiac muscle is uniquely designed to operate the heart, rhythmically contracting and relaxing for an entire lifetime without suffering from fatigue. Branching out the connective tissue section, we establish blood. Blood is a specialized fluid transport network, carrying red cells for oxygen, white cells to fight infection, and platelets to clot wounds. Finally, we diagram the physical connectors. Ligaments provide elasticity, connecting bone to bone so your joints can bend. Tendons provide tough anchoring, connecting muscle directly to bone so your skeleton can be pulled into motion. By physically locating these tissues on your own elbow and shoulder, you bridge the gap between abstract anatomical terms and your own physical mechanics. Look at your completed map. You have now organized the core foundations of biological tissues on a single page. This spatial layout consolidates multiple textbook chapters into a single reference point, allowing you to scan for specific shapes and functions during review. Understanding these structural shapes and rules on paper is the practical prerequisite for identifying real-world cell clusters on a glass slide. By structuring this complex data into a visual hierarchy, you are now equipped with a diagnostic key for analyzing both textbook diagrams and real-world specimens.